All right, let's focus on some of the positives in our lives, and that's learning about molecular biology. It's exciting. You guys are going to come out with full education. You're going to be aware of molecular biology issues and a lot of other things, so I'm hopeful with you guys, young people. All right, so was that enough pep talk to lift up the spirits? If not, I can tell you some bad jokes, but we can pass that. All right, so what we're going to do today is we're going to talk about transcription regulation, and this is a very important lecture for a couple of reasons. One is it's going to go through a lot of the techniques and show you how we know what we know about transcription regulation. And these are some of the techniques that you might actually use in the future, or you will be able to read a paper and understand what they're doing based on these techniques. Of course, there are a lot of variations over these basic techniques, but you're going to need to understand the rationale behind them to actually catch up with those variations, okay? All right, so why is transcription regulation important? It is the first step of gene regulation. Gene regulation can occur at multiple steps, like at the step of transcription, at the step of translation, or the protein stability, etc. And the reason why it's important is because the different types of cells in our body in animals express different types of genes and making specific proteins that are important for the specific cell structure and function. So what that means is the muscle cells are going to express a set of genes that are going to be important for its muscle cell structure and function, and that's going to be different from the nerve cells, right? For example, in this example here, the nerve cells are expressing this gene, acetylcholinesterase, and the tau protein, which are important for the structure and the function of the nerve cells. And the muscle cell is going to express acetylcholine receptor, so it can actually respond to the nerve cells, and tropomyosin because of the need for its structure. So how does this actually happen? It happens because that there are a set of specific transcription factors that are found in a given specific cell. For example, in the muscle cells, one of the most important transcription factors that determine the identity of the muscle cell is called MyoD. So this is a transcription factor. You can see here the crystal structure is known. It binds to DNA as a dimer, and you can see the DNA binding domain sitting on the major groove of the DNA. And what it does is it binds to several regions in the genome. These are transcription regulatory regions, promoters, and enhancers that control the expression of a set of target genes. And you can see that these set of target genes include myosin light chain, heavy chain, creatine kinase, acetylcholine receptor, and calcium channel, etc. All of these genes that are important for the muscle cell function. So you can think of this as a MyoD coordinately regulates many genes that are expressed in the muscle cells. And in addition to MyoD, there are other transcription factors that are important to transcribe some other genes that are important for muscle cells. But MyoD is one of the most important transcription factors. How do we know this? We know this because if you take a fibroblast, like a skin cell, and overexpress MyoD, what happens is that cell starts turning into a muscle-like cell. So this is one of the we call them master regulators. It's a master regulator of a muscle cell fate, right? It can convert even another cell if it's ectopically. Ectopic means improperly expressed in a different cell. All right. So how is transcription regulated? It's not just regulated up and down. So the genes are not just turned on and off, though we do see that kind of regulation quite a bit. So think of this kind of regulation like a light switch. You can turn it on and off. And there's also transcription regulation that can turn genes up and down, so it's kind of like a dimmer. Instead of completely turning on and off, you can adjust the expression level of a gene. And both of these type of regulations are important to make up the final transcriptome. The transcriptome is all of the transcripts and the kind of transcripts in a given cell. Okay? So far, so good? All right. So, gene regulation is mediated by signals that feed down to transcription factors. So there's going to be some signals that is going to, the transcription factors are going to respond, and that's going to allow them to bind specific sequences in the DNA at enhancers and promoters, and in combination with other transcription factors, they're going to determine the amount of transcription from a gene. And how do we know this? Today we're going to go through this. But one thing I want to point out here is that the numbers of sequence-specific transcription factors in a given genome, in yeast we see about 300 of them, in worms there are about 1,000, and humans about 2,000, is quite a big fraction of the total number of genes, right? So in humans there are about 25,000 genes, and about 2,000 of them are transcription factors. Now, this actually shows you that the regulation is a very big part of how the transcription factors are going to read and utilize the genome. It also tells you that one transcription factor cannot be dedicated to one gene, right? Otherwise, you would have to have 10,000 transcription factors for 10,000 genes, right? Actually, the transcription factors work in combinations regulating multiple genes. So each transcription factor is going to have a whole bunch of different, hundreds of different targets, right? So that's what the two concepts that you can get out from those numbers. All right. So to understand gene regulation at the transcription level, first we need to know which DNA sequences a transcription factor binds to, right? So now we're going to go through the techniques that will allow you to understand what DNA fragments a transcription factor targets, therefore which genes it actually regulates. All right. So the first question that you might ask is, which DNA, so you have a gene of interest, right, and that's being regulated by a transcription factor, and you might ask, which DNA fragments around my gene is important for its transcription, right? And the DNA fragments that will be important will be promoter and enhancer, right? So you can determine the promoters and enhancers in a gene. How do you do that? You do that using reporter assays, okay? So let's talk about what's a reporter. A reporter is a gene that's biologically inert, so that means it's not going to cause any problems when it's expressed in the organism under study. So, for example, if you're studying a muscle cell or like a human cell, one of you can use a reporter that is not normally expressed in that cell, and it's normally not present in that organism, and also when it's expressed, it's not going to cause any problem. And the reporter should be that the product of that reporter should be easy to assay or visualize, right? So give me an example for such reporter, because we talked about this, and Mark Siegel also talked about this many, many times. Give me some examples. Oh, what happened here? All right. What are the examples of some commonly used reporter genes? GFP, right? GFP is a protein. When it's expressed in a cell, it doesn't cause any problems, but does it have an easy way to visualize? Yes, because it fluoresces green, right? So it's like the easiest thing. You put on under microscope, and now you know. What other examples that you guys used? Beta-galactosidase. That's right, beta-galactosidase, right? So it turns X-gal into blue so that you can actually visualize the product of that reporter gene. And for detecting RNA, you can use in situ hybridization, which is a method that visualizes the presence of the certain specific mRNA that you're looking for. All right, so that's what you are going to be able to, that's what you're going to do. So what you are, the way to study then a certain regulatory sequence that you're looking for is, what you can do is you can put that regulatory sequence in front of, clone that regulatory sequence in front of a reporter gene, and if that regulatory sequence activates the transcription of the reporter gene, you're going to make mRNA of the reporter gene and the reporter protein, right? So if this DNA sequence is capable of activating transcription, you're going to see a signal. It's as simple as that. So how do we actually do this? So, for example, you're interested in the upstream region of this gene. So this is the DNA fragment that you're going to take and determine what region of this DNA fragment is important for transcription activation. Okay? So this is a large fragment. You want to know what little pieces are important. So what you can do is you can make that DNA and make shorter versions of that DNA using a technique like, what can you use to make shorter versions? Molecular technique, PCR, right? You can use PCR to make smaller fragments, right? So you can make smaller fragments of that DNA, and you can clone that DNA upstream of a reporter gene in a plasmid. So this is molecular cloning. You went through this several times, so I'm not going to go through that. So what you can do is you can take each of these DNA fragments and clone it upstream of the reporter gene. You ligate these, the plasmid vector and the fragments, and then you can transform E. coli and isolate E. coli that has each of the different types of plasmids, okay? And then you can purify your plasmid containing DNA, right? The DNA, the plasmid DNA. And now what you have is you have the plasmid containing different lengths of the fragment. Then what you can do is you can take each of these and transfect cells with each of these plasmids separately in different cultured cells, in the cultured cells, okay? So in the cell, what you're going to have is you're going to have a fragment that you're testing if it activates the reporter. And what you do is you can take these cells and prepare cell extract, or you visualize these cells, some kind of assay that is going to test the activity of the reporter enzyme. And then what you can produce is some kind of a result table like this, right? So for each plasmid, what you're going to have is is there a reporter gene expression or not? And the way that we typically do this is, if we do not have accurate quantitative measurements, we can use these kind of symbols, like the more pluses you have, the more expression you have, and the minus would be no expression of the reporter, right? So for example, this plasmid number 1 supported high transcription of the reporter gene, but the plasmid number 5, which has the smallest fragment, did not support any transcription. Is that clear? All right. So how do we then read and interpret the data? So what you will do next is you're going to put your fragments next to the results, and you're going to look which deletion has caused a problem, right? So when you had that larger fragment, you had supported high transcription. When you made a smaller fragment that lacks this region, you supported just as much transcription. So is this region of the DNA important for transcription activation? No. But when you went from the second to third construct, where you deleted this region of the DNA, you went from having lots of transcription to very little transcription, right? So then you put a little box here saying that region of the DNA is important for transcription. And then you went from that to losing this piece, which made no difference. But when you lost another piece in addition to what you lost here, you basically eliminated all transcription and you say, okay, that's another region that supports transcription. Is this clear to everyone? All right. So this kind of analysis has been done a lot for identifying enhancers for a given gene. Okay? So what researchers have done is they took multiple DNA fragments. For example, here's a gene with all the exons shown in red. The exon 0 here is named 0 because it was found after the gene was annotated. So they didn't know that this existed. So they corrected their annotation. That means this decision of where the exons are. That's why it's named 0. And so all of these fragments that they have used supported transcription of a reporter gene in an enhancer assay. Okay, enhancers are actually different from promoters. Okay, they work in a modular way. That means that each enhancer drives the expression of a gene in a given set of tissues or in a given developmental time point. Okay, so they do not support expression in all of the tissues at all times, but each fragment is specialized to drive the expression of the gene in a certain tissue. For example, this fragment here, this enhancer here, was found to drive the expression of this gene in pancreas. And how do the researchers know that? What they do is they take each of these fragments and clone them in front of a basal promoter. A basal promoter is a promoter that can support transcription. So it has all the required basal DNA sequences that you will find in a transcription promoter region of a gene. For instance, give me a DNA sequence element that you find at the promoter of a eukaryotic gene. Box, right? So it would have all of those DNA sequences, so it can support transcription. But by itself, it's a weak promoter, so it's not going to support transcription all that much. So if you have only this basal promoter, LAGZ is not going to be expressed very much. Okay. But then what you do is you take your DNA fragment that you suspect might be an enhancer, you clone it upstream of the basal promoter, and then what you do is you express make a transgenic mouse or inject in the mouse, etc., right? And now what you can do is, where is this gene expressed? For example, you see that it's expressed in this region, and then two enhancers shown here support expression of that gene in the retina, so you can see the expression in the retina, right? So you can see that the different DNA fragments support the expression of that gene at different tissues. So enhancers are also different from promoters because they're not position or orientation dependent. We talked about this already at the prokaryotic enhancer. What was the example there? Which regulatory system did we see an enhancer in prokaryotes? Two-component system, right? Where the enhancer position or orientation was not important for its mechanism to activate the gene. All right. So, okay. So you can find these DNA fragments. Sometimes they are quite large that can support transcription as an enhancer. But you want to narrow down the region that is really acting like an enhancer. The typical enhancer length in humans is about 400 base pair or so, right? But you can't start, if you have a gene of 50 kb, right? And you have DNA fragments that you wanna test, you're not gonna take that 50 kb gene and divide it into 400 base pair regions. Then you're gonna have to test thousands of constructs, right? It's too much. So what you can do is you can take a larger region, and once you determine that can support transcription, you can narrow down. You can step in and find out which region is actually important. All right, so how would you do that? What you can do is you can do the deletion assay. just like the way that we went through a couple of slides ago, what you can do is, now that you know this fragment is working as an enhancer, you can make smaller and smaller versions of it and determine what region of that fragment is important and working as an enhancer, right? Does that make sense? All right. All right, so I just told you guys that unlike promoters, enhancers are orientation independent. So given this experimental setup, how can you test this? What did the researchers do to find this out? Given this experimental setup on the right, shown here, right, with the basal promoter, the enhancer sequence, and the laxi, what would you do? You can invert it, right? And you can also find out if it's position independent. What would you do to do that?
Would you put it after the promoter?
Exactly. You can take it and clone it downstream of the laxi. Can you do it after the promoter?
No.
Maybe, maybe not, right? If it doesn't affect the transcription, that is, if it gets transcribed, but you're going to still translate the reporter gene with no problem, then it's not going to affect. But if it affects the transcription, then that is like, if it messes up with the transcription start site or something like that, then that's not a good idea, right? So a lot of researchers, what they do is, instead of putting it over here, they put it downstream of the gene. And the enhancers, the way they work is through DNA looping, so they can loop to the promoter from the downstream of the gene, and no problem, they can still work. Any questions so far? All right. Okay, so now these techniques, the deletion reporter techniques, is going to give you a certain DNA fragment that you know is important. And these fragments are typically large because it's very difficult to make many, many, many small deletions. Then you'll have to do many, many, many constructs, right? So, but it also doesn't tell you if the sequence there is important. What it tells you is that the DNA fragment is important, but it doesn't tell you if the sequence is actually important. How do you actually test that, right? So you ask the question, which sequences in the DNA fragment that I identified are actually responsible for activating transcription, right? And for this, the type of assay that you can use is called a linker scanning mutation assay. So what you do is you again have your the DNA fragment that you know is capable of activating transcription that is cloned in upstream of a reporter gene, and that actually makes the reporter expression, which you are going to measure in this case the TK mRNA. That's the reporter. All right, so next what you can do is you can make random mutations at different regions of that DNA fragment and clone each of these fragments upstream of the reporter gene again. So these little dashed lines here are showing you what region of the DNA fragment was randomly mutated. So that means you just incorporate messed up sequences there, okay? And the rest of the fragment is normal. And then what you can do is you can determine how that affected the transcription of the reporter, right? And so, for example, let's read this data now. What happened is that when you mutated this region here, you made no difference to the transcription. So that means the sequence of this region is not important, right? And then you made a mutation in this region, and what happened is the amount of transcription went down. So you say, okay, this region is might be important, right? And then what you do is you're scanning the sequence so you can make your mutations in regions that are overlapping, right? Continuous. And you look at the next fragment, and what you see is that also transcription is low, and you say, okay, this fragment is also important, right? But then you make a mutation a little bit downstream, and what you see is that this fragment, again, now this time has no effect on the transcription level. And now you know that the region that was important, the critical, was this region, right? This region, mutation of this region has made no effect, right? So by going down the data like this, you can narrow down which regions sequence are important. Is this clear to everyone? What I suggest you do is at home, take this slide and try to like do it yourself. Try to read the data yourself and see if you come up with the same answer by doing this. It's kind of like a puzzle. If you like puzzles, you're going to like this kind of data. Okay? All right. So through this type of assay, what the researchers could find is that the DNA sequences that are important for the transcription, which they call the control elements, are found here, here, and here, right? And so they can name them control element 1 and 2, and etc. Okay. Any questions so far? Yeah?
How do they know which fragments have been mutated where and how those are associated with transcription?
So they make the mutations. Right. So what you can do is you can PCR amplify the sequences so that you purposefully change the sequence in only one region of the DNA fragment. Exactly. So in this linker scanning mutation assay, what you do is, for example, you can take a 10 base pair region and jumble up its sequence within the bigger fragment, right? But same kind of principle goes even for single mutations. So if you suspect that there is a transcription factor binding site in one of these regions, and you know that for that transcription factor binding that a certain sequence needs to be there, let's say ATGC, and you can change it to ATGG, right, and clone that in front of a reporter and see how that affected transcription. So it is to your liking. You introduce the mutation, so you know what region you introduce the mutation.
So if the normal mRNA has like 3 plus signs, and you create a mutation, and like it instead of the transcription happens at like 4 plus signs, how would you read that?
What would you say? Nice question. Normally you see 3 plus signs. You make a mutation, you see whoop, things going up. That is totally possible. And we learned about these type of mechanisms in prokaryotic as well as eukaryotic transcription. Think like opera.
That like raise the mutation, like raise the affinity to like the mediator or activator?
That is, it could, not the mediator, because mediator protein complexes usually don't have sequence specificity in their binding because they regulate thousands of different genes, right, across the whole genome. But it might increase the affinity of a transcription activator, but more likely there is another mechanism by which this happens a lot, because activators are not the only transcription regulators.
It could decrease affinity to the repressor.
It could be messing up with the repressor binding site, right? Because the gene's transcription is not always activated by a transcription it's not always regulated by a transcription activator, right? It is also regulated by repressors. And in fact, the cumulative effect of activators and repressors in a given gene helps it turn it up and down, right? In lac expression, remember, in the condition where there was lactose but also glucose, you had a little bit of expression, right? And then when you bring in the activator, expression went up. So the two, an activator and a repressor, together had that up and down mechanism, right? It wasn't like the trip repressor where you had it on and off, because only trip repressor works on that. Does that make sense to everyone? So yes, you can have transcription activation, and more likely than not, the mutation affects the binding of a repressor, so it no longer gets repressed. Good question. All right. So next, what you can ask is you found some sequences that are important for the transcription, activation, or repression. And then, for example, you might suspect a certain repressor and activator binds to these sequences. So you wonder, where does my protein bind in the sequence? Okay? And the one assay that you can do to determine that is called a DNase footprinting assay. All right. So what do we do here? What you do is, when transcription factors and other proteins bind to DNA, if their affinity is high enough, they prevent something else to get in. Okay? So they have a footprint. They're sitting on something, so protecting that site. Okay? And that principle is used for a lot of these type of techniques. And in this technique, that principle is used to prevent DNase cutting. DNase is an enzyme that can create nicks in the DNA. Okay, so it can cut the sugar phosphate backbone, and it's an enzyme that is pretty much has very little sequence specificity. We can ignore it. It has no sequence specificity. It can cut anywhere. Okay, so what you're going to do is you're going to make a double-stranded DNA, a population of them, and you can make this by PCR or whatever way that you want to make, right? That contains the sequence that the sequence that you determined was important for transcription, activation, or repression, etc. And then within the sequence, there is a region that is bound by the protein, right? You don't know that green region yet. And what you do is you radioactively label one end of the DNA fragment. Give me a way in which you can do this, a PCR-based way. How can you label a DNA fragment, a double-stranded DNA fragment, on one end? Think of PCR. PCR is an amazing technique. You can imagine anything you want in the world. Imagine me a technique an approach that will give me this. What is that? More specific?
Would be a radioactive primer.
Excellent. What you can do is you can use a radioactive primer that's going to amplify from this region that's labeled, right? So everything that you amplified with that primer is going to be now labeled at that region. Okay? So that's one way to create these kind of probes. And that's the most common way people do this. All right, so now I shouldn't say this is a probe. This is not a probe. It's a DNA fragment that is labeled on one end. Okay, so now you have a population of these fragments, and what you can do is, let's imagine all of this is in a tube, and you put the DNase enzyme in there. DNase is going to go in and cut in all of these different regions, and you titrate the amount of enzyme such that it's going to create only few cuts per DNA fragment, okay? So what it can do is it can cut anywhere, right? All right. Now, you do the same thing in another tube in which you actually took the DNA fragment and first bound it to your protein, okay? So your protein, you used enough amount of the protein that you know most of the almost all of the DNA fragments in that tube is going to be bound. And when the protein binds to its binding site, it's going to protect that. All right? So now when you put the DNase into that tube, DNase is going to be able to cut here, 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 etc. Is it going to be able to cut here? No, because it doesn't have access to that DNA. All right? So now you have this fragment here protected from cutting. But then how do you detect that? That's the next step. All right. The way that you can detect that is that you can take all of those DNA fragments and now run them on a gel. So this is a long polyacrylamide gel that you can run. It resolves the DNA fragments of different lengths. Okay? So here is the profile that you're going to get when you have no protein that is, you're going to have lots of cutting, right? So each of these fragments are generated through some random cutting. What you're seeing here is the result of a denaturing gel. So what that means is you're going to see single-stranded DNA on that gel. What am I seeing here? The result of this cutting will be the DNA fragment that contains the labeled end. Because this is a radioactive gel, you only see the radioactively labeled DNA fragment. Okay? So this is running over here, and this is running over here, right? And this is going to be running over here, etc., etc. Does that make sense? Each of the labeled DNA fragment you can see on a gel. And the shorter fragments that are closer to the labeled end are going to run faster. And the longer fragments that are farther from the labeled end are going to run up high, right? Because in a gel, shorter fragments run faster and longer fragments run slower. Does that make sense? All right. Then here's a question. Are you going to see this fragment on the gel? No. It's there. It's not that it's not there. You're not going to see it because it's not radioactively labeled. You're detecting only radioactively labeled DNA. Does that make sense? All right. So what's going to happen? Then what you're going to do is you're going to compare the profile that you get from naked DNA to the profile that you would see when your DNA is incubated with the protein that you have at increasing concentration. So here, this DNA fragment was incubated with TBP protein, and this triangle basically tells you that the amount of the TBP protein that you added to the tube increased from one lane to the other. Okay? And what you can see is as you increase the amount of TBP protein, here is a region of the gel that you see is now being cut less and less. And what does that mean? That's the region of the DNA that is being protected from the cut, right? And you say, okay, here is the region that I know TBP is binding and protecting. Then what you do is you go to your ladder, right? And then determine where is that the region in the DNA. And the ladder is telling you that these DNA fragments are from 20 to 40 base pair. Then you know, okay, this is the region that is from 20 base pair for approximately. I'm saying for 20 base pair from that side and 40 base pair here. And the region in between here corresponds to this. Does that make sense? There is a study question that asks you to do it yourself. So once you do it yourself, you're going to understand it much, much better. And if you don't get it, just ask it during one of these sessions or the recitation, etc. We can cover that. But it's a problem set, so it'll help you understand that. Okay. So I had that question already, which is, are we going to see these fragments in the gel? No, we're not going to see them because they're not labeled. What you're going to see is the DNA fragments that are protected. For example, here is another protein. So it was TF2D in this case, was incubated with this DNA fragment, and what it binds and protects is that it binds over here, and it protects here, and then you can see that it protects here. But does it protect here? No, because even if you increase the protein amount, it did not protect those DNA fragments from the cut. And what you can do is you could go to the ladder again and then determine what regions of the actual DNA fragment was actually bound. All right. All right, so DNA DNA footprinting assay is going to tell you exactly what your protein binds in a given DNA fragment. All right, the next question that you might ask is, does my protein bind specifically to my DNA sequence? So sequence-specific transcription factors bind to anywhere between 6 to 12 base pair DNA sequences that are a specific sequence, right? And you might want to know if your protein is a such sequence-specific transcription factor that binds to that DNA sequence. We call them DNA sequence motifs, okay? So it's like if it's AT, GC, AC, whatever, that will be a 6 base pair DNA sequence motif that are bound by a transcription factor. And how do you do this? We do this using a gel shift assay. In this assay, I'm just going to give you what experimental condition is. You have a tube, okay, and you make DNA that contains the double-stranded DNA that contains the sequence that you want to see if it binds or not, if your protein binds or not. Okay, and what you do is you fluorescently label this DNA. It can be on one end or the other. It doesn't matter because this DNA is going to be kept as double-stranded. Okay, and then what you next do is you put your protein in here, and if your protein binds to this DNA, it's in a high enough affinity, it's going to bind here, right? And now you're going to have a DNA fragment that is bound to a protein. Then what you're going to do is you're going to take this thing and you're going to run in a native gel. This type of gels, what they do is you can run the protein DNA complex as a complex. It's not going to denature that. It's not going to break those interactions. You're going to see the whole thing running. Okay? Then you're going to use radioactivity to see the fragment sizes. Now, if a DNA fragment is not bound, it goes and runs through this gel faster. But if it's bound by the protein, it can't run as fast, so it gets shifted up, right? Does that make sense? Can you guys? So imagine you're the DNA fragment, okay, and I'm the protein. And if I bind you, I hop on your back. Like, don't try to visualize it. I hop on your back, and normally you would run really fast, but now you got me on your back, you're going to get slow. Okay? Same thing. Sorry for the analogy there. But, so that's basically the DNA fragment is going to get slowed and shifted up. All right, so to interpret that kind of data, you need some controls, right? So in any experiment, you need controls. So let's look here at some of the controls. What you're going to do in this lane is you mixed up your labeled DNA with a protein that you know doesn't bind to that DNA. Okay, so that's the control protein. And what you're going to see is you're going to only see the DNA fragment running pretty fast, so you know that this is the free DNA. Okay, and here you used a DNA fragment of sequence here. In here, what you did is you have your DNA, but you added no test protein. Okay, and it's going to run exactly the same place because there is no protein binding to the DNA. Now here you're adding your protein in increasing amounts. So these little rectangles are going to tell you that you're adding increasing amounts. And what you can see is that as you added a little bit of the protein, a small proportion of the DNA fragments are shifted. Right? So you had enough protein to bind to a certain percentage of the DNA there, so that you have a shifted thing. This is the free DNA plus your protein. And the rest is the free DNA that's not bound, right? And as you increase the amount of the protein, you had more and more binding. And the distribution of the bound and unbound DNA fragments is going to be determined by the how tight that protein can bind, right? So, all right, so now you establish that your protein can bind to this DNA containing that sequence. But it doesn't tell you if it binds to that sequence specifically, right? There are some proteins that are going to bind to anything. Name me one of those. Give me a protein that binds to pretty much any DNA in eukaryotes. Histones, right? They don't have much sequence specificity. They bind to pretty much anything. So in fact, if you throw some histones to this, it's going to stick like crazy. It loves DNA. All right, so how do you test then if the binding is specific? What you can do is you can use what we call a competitor, a cold competitor. A cold competitor is when you do your experiment, your DNA is labeled with some radioactivity, right? And your protein is binding to this DNA. What you can do is you can put the same DNA fragment, like here, this is the same DNA, but now don't have it labeled, okay? So this is the competitor. It's the cold. It doesn't have any label. Now, because protein to DNA binding is basically a biochemical event, right? It's going to have a certain on and off rate. And when you give them a lot of cold substrate, what's going to happen is that when this protein gets off, it's going to bind to one of the colder ones. And the more cold competitor that you put there, the less number of proteins are going to be bound to the labeled version, right? Does this make sense, guys? It's competing off, right? Okay, so it's competing off. All right, so when you add a specific competitor, that means the same competitor that has the same sequence of the labeled probe, what you're going to see is that your protein is now lost. That doesn't mean it's not binding to any DNA. It's just binding to the cold stuff that you don't see on a gel, right? But if you add a non-specific competitor, that means a DNA sequence, a DNA fragment with the sequence that doesn't include this, right? Then what will happen is that it's not going to be able to compete because even though there's lots of cold DNA, this protein only loves its own sequence. So these other sequences are not going to be able to compete the protein off. Does that make sense? Any questions about this?
How would the DNA bound to the non-specific competitor still show up on the gel if you weren't using labeled DNA?
That DNA that you see here is this, the unbound, right? Because in the tube, there's still labeled DNA. It's just not bound anymore. That's why you're going to see this. Is that what you're asking? Mm-hmm. Any questions? All right. So now you say, okay, I have this DNA fragment that's about that long, and I suspect that this is the actual transcription factor binding site that is shown in the capital letters TAATCC. How are you going to use this assay to test if your transcription factor is specifically binding to that sequence? And what you do is you mutate the sequence. So you make a probe, labeled probe, just like your normal experiment, but instead of having that sequence, you change it to something else, right? And now you mix your protein and increase the protein amount and mix it with that mutant probe, and you no longer see binding. And from this experiment, you can say that the my protein specifically binds to this sequence here, right? Does that make sense? Question about this? All right. All right, we went through this, which is that the mutant is going to allow you to test the specificity of your protein for your sequence. All right, so these assays that we went through, some of them, like the footprinting assay and the gel shift assay, where you look at where your protein binds, is in vitro. That means outside the cell, right? It's done in a tube. How do we actually map protein binding in vivo? Right? This happens to be one of my expertise, where I look at protein binding in vivo inside the cells. And I use a biochemical method combined with genomics to do this. Okay? So this method is called chromatin immunoprecipitation, followed by high-throughput sequencing, abbreviated as ChIP-seq. So what you do is, first you have a whole bunch of cells, so you have a population of cells in a dish or whatever, right? And what you do is you cross-link the protein to DNA, so cross-link the chromatin with a chemical called formaldehyde. Formaldehyde is small enough to go through the membranes, and what it does, it chemically cross-links the DNA to protein and protein to protein. So what it does is it takes like a snapshot of whatever is happening in the nucleus when you add that formaldehyde. And that's done across a population of cells. Let's imagine now all these little circles that are drew completely horribly are the nucleosomes in a given chromosome. And this is the same chromosome across different cells. And then let's say this is the triangle is the transcription factor that binds to a certain site. Now, transcription factors in a given population of cells, in a given cell, it has a certain probability of binding to a site. Okay, and if that site, the transcription factor has a high affinity to a certain site, when you look across different cells, you might see that the transcription factor is binding to that same site in every cell. Does that make sense? But if the transcription factor has lower affinity for a second site, sometimes it's going to be on the DNA and sometimes it's going to be off the DNA, right? It's an equilibrium. And when you look at a population of cells, at that site you're going to see that transcription factor will be bound at some of these, in some of these cells and not the others. So when you add the formaldehyde, you take a snapshot of that binding. Does that make sense? All right, now we're going to find out where these sites are. So to do this, what you do is you take your formaldehyde cross-linked chromatin and you shear it by sonication into smaller pieces. And what that is going to be used for is that it's going to be used for an immunoprecipitation assay. Immunoprecipitation is precipitating the DNA or whatever you want using an antibody. Right? So what you're going to do next is you're going to use an antibody that's specific to your protein of interest, in this case the triangle. So I use an antibody that's specific to the protein, and the protein, some of these proteins are cross-linked to the DNA, right? You can see that DNA, that's red. Now, that is my immunoprecipitation, which the shorthand version is IP. That's my IP sample. It should include all the sequences that are bound by my protein. And now I'm going to take a reference sample, which is the control sample, where I'm not going to do any immunoprecipitation. So it should have all the DNA that is isolated from the population of cells, including the ones that are bound and unbound. Right? All right. So next what I do is I reverse the cross-link. So formaldehyde cross-linking of DNA to protein is reversible at high temperatures. So I incubate my samples at high temperature that reverse, and then I can isolate the DNA from each of these samples. So I'm going to recover the immunoprecipitated DNA, and I'm going to recover the input or reference DNA. Same thing. All right, so I have these DNA fragments now, and what am I going to do with them? What I do with them is I take them and construct an Illumina sequencing library. Okay, what that means is that we're going to take each of these samples and push them through a set of molecular biology steps. So this is shown for one DNA molecule here, but of course is going to be happening in all the molecules in the tube. Okay, so the sonicated DNA fragment size is usually about 200 to 500 base pair. So the first thing that we're going to do is we're going to add an A nucleotide at the 3' end of this DNA fragment. So a cocktail of enzymes, what they're going to do is they're going to repair the ends, and they're going to add an A nucleotide. So they're going to add the A nucleotide at the end of the DNA fragment, which will be used to anneal and ligate that DNA fragment to a adapter sequence. Okay? So adapter sequence is this. These are short, double-stranded DNA molecules that we make, right? We artificially make, and we know what the sequence is. Okay? And what we do is we make sure that there is a T overhang here, and when we ligate that, it is going to specifically ligate to these ends and form this bigger DNA fragment, right? And now what happened is that I generated a larger DNA fragment where I know what the sequence is at the two ends, right? And this is important for the next step. But here I want to ask you a question. Why are we adding that A tail? We call it an A tail to the DNA fragment before we ligate. Why can't we do this with basically some blunt-ended fashion with the adapters? This is a more clever way of doing this, and there's a very good reason for that. Imagine what would happen if you didn't do that step and you used blunt-ended DNA fragments to anneal. Blunt-ended adapters.
You could find out either ends, like any orientation or order. You do not know what the sequence is. If it's like this one's supposed to be here, it could be on the other side.
Well, this A-tailing is done on both ends and on both adapters, so you actually, the orientation, you already don't know, right? Because this can go over here, or this can go over here. There is no sequence specificity. What else?
The blunt ends might anneal to each other.
Yay, excellent. The adapters may anneal to each other and form adapter dimers, right? And this is a typical molecular biology type of optimization. When we make protocols, we optimize things. Optimizing things, so you can think of this like engineering molecular biology. You have a starting material. You want a certain product, right? So you do everything here to make sure that your product percentage is as high as possible. But if you didn't have the A-tailing, right, and a lot of your adapters just stuck to each other, then you will have a lower percentage of your product, which will be not desirable, right? So here's one example in which molecular biology is used to create the product that you want by optimizing the approach. All right. So next what you have is now you can use, because you know the sequences here, what you can use is you can use primers that are going to anneal these sequences so that you amplify that DNA fragment. So what you have is you have a single DNA fragment here, went through a ligation-mediated adapter. You put ligation adapters through a ligation step, and then you did a PCR, so you amplified that. Okay? So this is one way to amplify a DNA that you have no idea what the sequence is, right? Because if you don't know what the sequence is, you can't amplify it. So this is one way to do that. So why do we need that amplification? We need that to generate the DNA fragments that are going to anneal to the Illumina sequencer flow cell, right? You guys learned about this with Professor Siegel. So those adapter sequences, those red sequences that are shown here, what they do is they contain the sequences that are going to, this is the flow cell, which has all these oligos here, and your DNA fragment is going to be able to anneal here because these oligos contain the complementary sequences to this adapter, right? So what you can do is you can take that and you can anneal it to your Illumina flow cell, and now you get the sequence of the DNA. Does that make sense? All right, good. All right, the sequence that you're going to get from this is that you're going to get a ton of millions of sequence reads from your chip sample, so that's the immunoprecipitated sample, and your input, that's the reference sample that you did no immunoprecipitation whatsoever. And the reads that you're going to get out of an Illumina machine are usually short, and what they're going to give you is they're going to give you the sequence read that is corresponding to each end of the DNA fragment that you basically used, okay? So you're going to get the sequence from 50 base pair of that end and 50 base pair of that end. All right, then what do we do next? What we do next is we want to know where do these sequences come from, okay? So we align those read sequences to the genome. So this is your genome. It's kind of just basically a line, okay? And you want to know where your protein binds. So think of the y-axis as where your protein binds. The more you see, the more protein binds. How do you do that? What you do is you take your sequences and you align it to the genome. Aligning means where in the genome is your sequence coming from, right? So you scan the genome like BLAST. You've done BLAST in your RIS stations, and find out where it comes from. And then you know that there are two, if you did a paired end read means you read on both ends. What you know is that the DNA fragment that was initially in the DNA, in the tube, was this DNA fragment that connects the two reads that you got. So what you do is you basically align the read to the genome, and you have many millions of reads, right? So you do the same thing across the genome with all of the reads, and so they basically align to all of these sites. And then what you do is you count coverage for each genome base pair. What that means is that you go one by one at each coordinate along the chromosome and say, how many DNA fragments that are aligned here that cover the space, right? Here, the coverage that is from the chip is 1, 2, 3, and 4. What's the coverage here? 1. What's the coverage here? 2. So what you can do is then plot the coverage along the whole genome, right? And that's plotted here. So the more coverage you have, you know that in the chip sample you had more of that DNA fragment. Does that make sense? Okay, so how does the data actually look like? Here's some data from my lab. So we work on a protein complex that represses transcription of the X chromosome in C. elegans, and it contains multiple subunits. One of these subunits is called an SDC1, sequence-specific transcription factor. It has lots of zinc fingers that love binding to DNA. And what we did is we did the ChIP-seq analysis of SDC1, and here are some replicates. So when you do an experiment, you have controls, and you do it many times to make sure it works, right? And here what I'm showing you is data from 3 replicates across the genome here. This is I can't remember, about a 20 kb region of the genome. And what you can see immediately is there is a site here where the protein is binding very at high levels, right? And then you look at the same coverage of the same region and look at the control coverage, you can see that there is no specific enrichment of the protein over there. Does that make sense? Any questions? All right. So this is how you know that where the protein binds in vivo. Okay. And what can you do with this information? Now, when you do a ChIP-seq of a transcription factor, because transcription factors bind to many sites across the genome, what you can do is you can take each of these sites and determine their DNA sequence, right? And so here's one binding site number 1 and 2 and 3, etc. And you know what the sequences of these sites are. And then what you can do is you can go to a bioinformatics program and ask, what are short DNA sequence motifs that are found commonly in these binding sites? And what it is going to spit at you is it's going to spit at you some DNA sequence motif that it finds. And in our case, this is the DNA sequence motif that we think that the SDC1 is binding, right? Okay, so you can find the consensus sequence among these binding sites. Does this data actually show that SDC1 binds to this motif directly in the genome? what does ChIP-seq actually measure? Does it say that this protein is directly binding to the DNA? I told you that formaldehyde cross-links protein to DNA and protein to protein. Now think back in the first step of the ChIP-seq protocol. What if STCT1 is not the actual protein that binds to the DNA, but it binds to another protein that binds to the DNA? Are you going to see STC1 sitting at those sites using ChIP-seq? Yes, because STC1 will be, let's say, STC1 binds to some other protein that directly binds to this motif, right? And when you do a cross-link, because it cross-links protein to protein and protein to DNA, when you use an antibody against the STC1 protein, what you're going to get is you're going to get these DNA fragments. Does that make sense? Okay. Now, we established, but we found a nice sequence motif that maybe it binds, so that's a really good start. Now I can ask the question, does it actually directly bind? What technique can I use to test this? To see if, what do I need to do to test that question? If it binds directly or not. That still is not going to answer the question very much, because if this interaction between STC1 and that adapter, whatever protein is binding, is strong enough, and your immunoprecipitation is going to bring it up, you'll still see it.
Is there a way to get it to have no affinity for protein but only for DNA to change the conditions? So it would only be bound to...
But then you would have to know what the nature of the interaction is, right? But you don't know. So think of some other experiment that we talked all through the lecture.
Can you do a gel shift?
I can do a gel shift experiment. So what do I need? I need a recombinant STC1 protein. I need to make that protein or purify it out of the cells. And then I need to make a DNA fragment that contains this motif, right? And I'll do a gel shift, and I'll mutate the motif and do a gel shift. And if it binds specifically, it should shift the DNA probe. And if I mutate the motif, then it should not bind it, right? Does that make sense? What's another experiment I can use? Gel shift is the first thing that researchers do right away when they suspect something like this. What other technique can I use to test for direct binding? Think back, just stroll through the slides from this lecture. Go back. Speak up. You don't know?
She said gem cell.
The GEMSA is exactly, that's the gel shift, right? So that's the first one we do. What other one? DNA is one, footprinting, right? If I mix my DNA with the protein and I see that it protects it from cutting, that I know, okay, it's like in this tube I only put my protein, I only put my DNA. If it binds, of course, it's going to protect it, right? So these are the type of techniques that you would do. The highlight today is just some data that I took out of my lab. For example, here what we're looking at is we're looking at about a 50 kb region in the C. elegans genome, and we have chipped various proteins. So these are a couple of proteins that we chipped. These are condensin subunits. These are SMC complexes, remember, that mediate chromatin loops. And here is the binding profile of RNA polymerase II. So we did chipped RNA pol II in these cells. And here's a negative control where we're not chipping anything. Okay? So what kind of information does these type of experiments give? For example, if I'm interested in certain regulation of proteins in one tissue type versus the other, what I can do is I can chip RNA pol II in one tissue type or the other, and I can see where it binds, and then I will know which genes are expressed or not expressed, or where polymerase II is at. right? So, for example, how do I know which gene is transcribed here? Is this gene transcribed, BET2? Yes, because pol 2 is binding from its transcription start side all the way to its transcription end side. Is which gene is bound? Is this gene transcribed? No, because you don't see much of pol 2 at all. In what regulatory regions do condensing subunits seem to be binding? Look at the pol 2 profile and look at the condensing profile here. Do you think this is an enhancer or a promoter? Is it at the beginning of the gene? Is it at the intron? Is it the end of the gene? The beginning of the gene, right? So that's where the promoter is. That's where the transcription start site is. So from this, what I can see is that my condensing proteins preferentially binds to the promoters of the genes, and I know that it represses transcription, so I can hypothesize that perhaps the condensing proteins, by binding there, the way they repress it is maybe because they interfere with the initiation process, right? Does that make sense? Okay, so that's the type of information that you can get. All right, the lecture is done for today. Good luck on Friday. Work hard. And I'll see you on Monday.